So, welcome everyone to today's talk titled Environment and Health, Open an Era of Environmental Discovery with the Exposome series, targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope the series will inspire international scholars, researchers, farmers, and business in the speaker, Professor Chung, who is an exposure biologist with a PhD degree in environmental health sciences. For exposure biology, it's quite a new area to me. He did to reactive chemicals. After graduation, Professor Chung received training in exposomics at the National Taiwan University and the Duke Kunshan University after further sciences at Harvard Medical School. His central research goal is to develop and apply measures and computational methods to identify the elusive environmental drivers of diseases and discipline the exposure disease continuum. Professor Chung has published in leading environmental journals such as Environmental Health Perspectives and Environmental Science and Technology, and was interviewed by news and magazines, invited to Sweden to conduct a technology transfer of one of his innovative assets. In addition, he has served as a grant reviewer for the Medical Research Council, United Kingdom, and as an architect at Planetary Health and Environment International. In this talk, Professor Chung will introduce the idea of totality of exposures, the exposure, recent developments in achievements of the field. He will also compare major measurement approaches and highlight key demonstrative studies. It is hoped that ultimately this effort will accelerate the pace of translating public health and preventive medicine. So without further ado, let's welcome Professor Chung.
Thank you, Professor Lam, for the introduction, and thank you for everyone coming this talk. So today I'm going to talk about something about environment and health, and then at the end of the talk, so I will talk something more about the application of the methods to perhaps like animal science and plant science. Okay, so just want to introduce a little bit about myself first. So I just recently come back from Boston and joined the Chinese University of Hong Kong at the School of Public Health. So before I came here, so I was mostly like biomedical and environmental health data scientist. And then right now, I have more like a flexibility about my own research. So I'm going to do two things. One is more like the wet lab research, and the other is more like the dry lab. And I'm hoping to integrate some of my knowledge and the research experience into something that could be helpful with also like a groundbreaking and kind of like a broad impacts to the research community. So for today's talk, so I'll give you a brief outline. So I'm going to have like 5 different parts of my talk. So the first one is about why we care about the environment. And the second one is about the recent development to study the exposome. And then I will also talk about the key approaches, measure the exposome. And then finally, about some development from my groups, and then a summary and a final remark of the talk. So let's talk about the first thing first. So why do we really have to care about environments? So then for this question, so we can also ask something more specifics, like a question about what really affects health and diseases. And then to answer this question, so we can have a very simplified like a formula, like what I show here, P equal to G plus E, and the P is called the phenotypes, and the G is for the genes, and then the E is environmental exposures. For example, like dietary exposure, pesticides exposure, and other smoking or our unintentional exposures. And in these models, so we have different interaction too, such as the gene and environment interactions, or the G and G interaction, or the E and E interactions. And then so we may wonder for this formulas, we have the G, we have the E. So why not we start with the G first? And for the G, so our first question is to ask, how well are we measuring the G? And for this, so we may also look at the some of the achievement first about the genomics. Say a paper in Nature in 2001 talks about the initial sequence of the human genome, the first draft of the human genome. And for that one, after these significant achievements, so we have a like a groundbreaking development in other measurement technology, such as the next generation sequencing technologies. And then we also have some other projects after this Human Genome Project. That's the international HeadMap projects that has an aim to understand the variation of the genetic sequence in the human population. And then with the mature and the lower cost of the measurement technologies, so we can do something called the genome-wide association studies, GWAS. And for this study, so we first need to have two populations. Like one is the population of the disease, and one is the control, they don't have the disease. And then we collect the sample, and we can use the different genotypes methods, such as using the snip away and doing some kind of measurements of key SNPs. Or recently, we have these next generation sequencing, so we can do the whole genome sequencing methods. And then with all this data, and then we can run the analysis, statistical analysis. So in the figure on the upper right, so I'm showing you a what they call the Manhattan plot. And it's basically trying to show you all the association that they were testing in the GWAS. So typically more than 10,000 K or 10,000 SNP, if it's like a SNP away. And each dot represents a SNP. And the y-axis is the p-value. They just make it like a minus not 10 to make it easier to represent what p-value we know as a positive number. And then you see there's a dashed line. And what that means is when you find a dot that is a SNP that is higher than that dashed line, that means they are statistically significant. And with these informations, so we can do some follow-up investigations, such as looking at the linkage disequilibrium. We figure out the a plot of SNPs that has high correlation with these only significant SNPs, and then we can do some file mapping analysis to figure out the causal genetic determinants of a disease. And then, last year I was at Harvard and I attended a precision medicine conference. It was held by the Department of Bioinformatics. So they invited one person, his name is called Jimmy Olahir, to the conference, talk about, share his experience. So he was a sickle cell disease patient, and he has to go to the hospital like every month to do some medical treatment of his disease. And one day when he read the newspaper, it's like there was a report about a medical institute. They were doing some kind of like a trial, new method about gene editing to cure the sickle cell disease. So he is kind of like very interested in being like one of the test subjects. So he approached the investigator and talked about whether he can join the trial. And finally, he has a chance to join the trial, and talks about his story about how he got cured, and kind of like I'm surprised. It's more like no side effects. It was like completely healthy, not like the previous gene editing technique before the CRISPR-Cas9 that I can often read some medical journals say have some editing techniques, but then we have strong side effects, have to stop trial. And for his treatments, I read the detail a little bit, so it's kind of and stuff. So he has to go to the hospital, and then there's some kind of medical procedures, and he has to sit down there like 8 hours, and then take the stem cell out from him, and then they will send the stem cell to a lab to do the CRISPR-Cas9 gene editing. That they will switch off particular genes that we know it is related to sickle cell disease. And then after this gene editing of his stem cell, so he has to go back to the hospital, kill all his stem cell, and then they have to infuse the edited stem cell back to his body. And then that was really kind of amazing things. Sounds simple, but it's a more like a painful kind of like the treatments. And he's kind of happy to share his experiment in the meetings. So there's a lot more like a pipeline to a dream that a world without the illnesses. Like so we have the sequencing technology for the genomes, then we have the human genome project, and then we have the sequencing technologies, and then we can apply that to run GWAS to figure out the possible disease causing SNPs on the gene. Then we have the CRISPR technique to do the gene editing. And with that technique, so we can fix some genetic errors, and then we can cure the disease, the genetic disease. So these figures show you the heritability of traits and diseases. So the x-axis is the heritability. So the greater the heritability means something is more determined by the genetic influence. So you can see at the very bottom of it, it talks about eye color. And then the heritability is almost close to 100%. So that means what determines your eye color is kind of like purely genetics and nothing about the environment. But then if you look at this figure, like the top half of it, so we have many chronic diseases like cancers, type 2 diabetes, stroke. And then these diseases, they only have about like maybe less than 25% attributions to heritabilities. And then we have quite a lot of known, we don't know why. And we can talk about these things are non-genetic factors, and they are likely mostly caused by the environments. And so there's a saying, they call it genetics loads the gun and the environment pulls the trigger. And what that means is it represents a disease model that the genetics gives you the susceptibility or confers you some kind of resistance to the disease. But actually, whether you or me, whether we have the disease or not, is trigger is determined by the environment, what we expose. And then from the figures, we can kind of understand the study of the environment is underrated. And we know the disease burden from that figures, many common chronic diseases. So it has a pretty high environmental factors, but not really from the genetics. But then the slides that I showed to you previously, so we know we are very good at measuring genetics, studying genetics, even fixing genetic disease. So we have very good analytical capabilities, but not exactly for the environment. So especially for the college, in my view, they kind of very traditional. And that was really kind of something surprise to me when I just joined the field. Because when I talk about measurement environments, the first thing in my mind is about doing objective chemical measurements. But in their view, it's more like questionnaire. So ask your subjects some questions. How many drinks do you have? Is take something like that. And then so let's go to what exactly is the exposomes. So the exposome is a framework to do systematic study of the environment. And it was founded by Christopher Weil in about 2005 from his commentary. And actually, at that time, no one really cared about that paper because on my memory, around that period of time, you can always find some commentary perspective that someone studies something, and the suffix home, and then they talk about some kind of home. And it's kind of like one of those, so nobody really care about that. And he defined that the exposome on kind of a very simple way that a life course environmental exposures, everything. And then life course exposure is kind of a concept, doesn't really mean anything practical. So it could be measurement per second, minute per hour per day. So it doesn't really like for a regular person, maybe 80 years lifespan. So can't really do that measurements. So people suggest that we can take snapshot measurement in important different life stages, like the infancy, childhood, adolescent, and adulthood. and then that is some kind of representative measurements. And then, around that period of time, I actually accidentally discovered his paper, I think about 2009 something, because I was searching what exactly meant by the exposure biology, and Google saw up this paper. And I think around in parallel, my mentor, Steve Rappaport, he also discovered the paper and talks about these exposome things, and he wrote a commentary in General Science, talked about have like top-down, bottom-up strategy to measure the exposomes. The bottom-up is more like you collect the sample, soil sample, air sample measurements. But he prefer more like the top-down approach because it's easier. You just collect the blood samples, and then whatever you found there is really exposed, like dietary stuff, maybe exercise, maybe some drug exposure, unintended drug exposure, like the livestock, the biotics. And then after that, he also published both papers about the exposome in journals to different audience, and this exposome concept really kind of like started to gain the momentum and the tractions. And then a few years later, and then another important person, Gary Mina, he talks about the exposome again, but he's more like redefining the exposome because he's a toxicologist. So he talks about not the exposure when you expose, but the biological response and the cumulative measure from that exposure. And actually, his definition has a pretty profounding influence, because right now, like half of these exposure study is talking about the biological responses. They're not looking about the exposure. And then, so Gary and Morel, they recently also have a review paper in Science talks about exposomes. They made a more operational exposome. It's more like whatever you can do, whatever you can measure, and integrate them. This is especially kind of things more prominent in the European project. They combine different existing study measurements, ecosystem, green space, lifestyle, smoking, social stuff, your agent, chemical radiation, that kind of things. Measurement can be from remote sensing, questionnaire, personal sample, like the variables, like chemical measurements. And then the exposure research paradigm is kind of you ask these leaders, they have consensus, and I talked with them like a few months ago. No consensus, but my understanding is some has tried to study integrate the environmental exposure, trying to things more than one at a time. So I would say it's more like a four is exposure research paradigms. And in addition that you will look at the things like people at different life stages, or you look at molecular or high level measurements, or you try to tackle multiple comparisons that you don't really interpret the p-value the way you do traditionally your statistical tests. You have to do some probability corrections, which is very common in GWAS studies. And then the research paradigm right now is going to a direction like so we have the exposure, like the figure on the left-hand side, and then we have the outcome, on the right-hand side, and then we have different omic stuff to reach the study to try to understand the association about exposure to disease. Whether we can understand discover certain mechanisms that really connect the association. And then let's talk about some recent development of the exposomes. So because it's really broad, so I will only give you a overview some kind of like a holistic questions about what we're doing. So about the government, so they do have put these concepts in strategic plan, like the NIEHS Institute of Environmental Health Sciences. And then they in Europe, I'm not that familiar about the policy there kind of things, but I know they have the Horizon 2020 funding program to provide funding for the European project during this kind of exposure things. And then in the US, so we have the NIH special application, the kind of funding notice. So EBA is kind of asking you to submit like a grant to check eco mixture. And then we also have resources like this PubChem, a database for you to look up your chemicals, some information about the spectrum, the kind of things. EVA, they have their own databases. Tox is one of them. So they do have things like the exposure pathway, I mean, the source of the exposure. And then we also have the here is more like a resource for doing measurements. So it works like you have a sample, you want to apply for the measurement, and then if they approve it, so you don't have to pay, you just send a sample and they will do the measurements for you. Things like maybe like pesticides, PFAS, and trace elements that are typically found in food or the agrochemicals, agro industry. And then we also have things more like oxidative stress, metabolomics. And then we have kind of like important biobanks, like the UK biobanks, 500,000 people have very rich genetic information, also the phenotypic informations. In the US, they have the all of research program, double the size of the UK biobank. Actually, they're trying to do that. I'm not sure whether they can really do that because this is more like the second kind of like report of the project. Because the first one is more like a coming from the Obama. They have the precision medicine initiative, and then they figure out the original plan is too expensive, not possible. We put and we start everything, and we have this first research program. Then for the another government institutes, so they have their top map program because they focus on these, and they were funding the cohort PI to genomics, metabolomics, also exosomics. It's really nice. You have different databases, but sometimes in the government it don't really work. Like one time, my PhD mentor, he tried to talk to a PI of a heart disease cohort here in this top map, and he's interested in exposomics. So we talk to here program director, like we want to apply and do something, and then they approve it, so we can set the sample and do some measurements. But then the things comes from when we need to share the data, they want us to share the data in the platform, and top map they want us to share the data in their own platforms. So we couldn't sort it out. So about this proposal, this project. So there's some kind of bureaucracy here. And then in the industry, so we have different companies, say on upper right, so the my exposome doing personal environmental monitoring, and then some company metabolomics. And then my PhD mentor, Chung Patel, so he has his own AI startup company about doing geomapping and environmental health. And one of my classmates at UC Berkeley, so she was actually a postdoc at Stanford, and somehow just in the exposome topics, and then she pitched the funder in the Y Combinator that she was later able to get the startup and has a million market company to do these exposure measurements. And then now we just go back to the academic view about some kind of interesting measurement developments. So in a Google Map, in Google Map you can have a little yellowish human figures. Drop it, and then you will have a street view. And that street view is coming from that you see there's a car on the lower left that they have the camera just circle around. You see the picture, and that's the data come from for the street view. And then so they have a project that they think they can install a real-time air pollution sensor. So along with the camera, they circle around the city and they do some real-time measurements of the air pollution. And then one of the investigator in Netherlands, Remnants. So he tried to merge the typical model, land use regression model, to estimate air pollution with these high resolution real-time measure of map. And then he was able to create some higher quality air pollution map by fusing the data together. And then there's some development about sampler, like a micro needle patch to collect the biofluid. It's from the Science Translational Medicine Journal. It's a little bit tricky to understand what they were doing, because when I read the original paper, I just get the figures. On your right-hand side, and like some needles, and then some like a circular skin Then I have to kind of like go to the supplementary and try to understand what exactly they can do. It's more like they have the microneedle, that kind of device, and then they approach your skin, and then they then have a vacuum pump to extract the liquid in the cell, and then they will do some measurement using that liquid. So they may provide a video, so I have to touch a look and understand what they were actually doing. So they have some kind of plastic the skin, and then they have something like a comb, but with a microneedle, and it keeps tapping the skin. And later, they will suck out some liquid out from that kind of like a micro patch, that kind of fluid. Then we have something called the exposometer from Michael Snyder, and he's the one from Stanford, and he likes to do this kind of multi-omic stuff. And he got something like exposometer. It's actually not the things that he developed. It's like from the RTI that company, and they have these personal exposure monitor for measuring air pollutions. So they just modify that to have a filter to collect the biological component in the air, and also have some kind of stoppants to absorb the VOC, semi-VOC in the air. Then say after 2 weeks of sampling, they will take out these sampling fibers, stoppants, and then do the next generation sequencing and also mass spectrometry and the exposure. So another paper from Michael Snyder that really likes this kind of multi-omic stuff. This time he tried to use a micro sampling device to collect blood samples. That say we have a couple of person collect the blood samples, and then they have some kind of a milkshake dietary bench exposure, and he keep collecting blood samples like maybe after regular intervals. And the innovative thing here is that they are using a device called a mitra that can collect like a few drops of blood that will be much easier than on the puncher. So it's more like you have type 2 diabetes. You just put your fingers, you have a couple of drops of blood, and then you can have this device to collect the blood, and then send it back to the lab for the measurement. And then for the analytical side, so I think my PhD postdoc mentor, Patel, he is one of the really pioneer here, because he's the first one to develop the methods exposome-wide association study XRAS, try to look at these totality of exposure in other databases, and try to reproduce some kind of analysis like we use in GWAS. And he was able to figure out that using this approach, we can detect some reproducible exposure related to type 2 diabetes. And I'll say this is really one of the key papers to help promote these exposome concepts to kind of research paradigms. Because at that time, people just talk about the concept, but they don't really have an idea how they do that. But then you have these extra analytical methods, and these combine different measurements, and then you can use this method to do some analysis. And then to understand like a lot of correlation from this exposure, so we have the exposome correlation groups that I and my mentor developed these kind of informatic methods. So it's basically like we try to show the correlation line between different chemicals. So the red one means it's a positive correlation, and then green one is like negative. So the darker the color, the thicker the line is stronger the correlations. So you can see very simply that we have the PCB, we have very high intra PCB correlations, have these red lines in that class, and also some kind of a strong correlation between CB and OCD organocholine pesticides, because they were both things more hydrophobic, more like kind of like a sticky compounds. And then for those more hydrophilic stuff like the metals, so they have another kind of like so in bottom half of the figures, and then we have another like a cluster of correlation among them. And then using the same study, I also did some kind of exhaust analysis, like 130 chemicals, and then also using the Manhattan plot to show the result. So the x-axis is more like the typical Manhattan point that we have the chemical exposure. Dot is exposure, and then I just color them by class. Like on the left-hand side is dot is one chemical, and then the y-axis is still the value, not minus not 10 of the p-value. And then the interesting thing is that if we don't adjust the p-value, just set the threshold at 0.05, and that will represent a line at 1.3 in this figure. And what that means is that if I don't adjust for the p-value, I will see a full of PCB and other chemicals. They were associated with a fertility endpoint. And then if we adjust for these multiple comparisons, then say using the bufferonic correctness, and then you see there's a line, red color on the top, and that's where the threshold comes from. And then you will be able to see there's none of them, they are pretty significant. And then based on these negative results, so we just think about what are the sample size needed for this kind of analysis. So we know we have to calculate sample size and power analysis when we submit a proposal, like a typical one at a time kind of like hypothesis-driven study. But for this kind of discovery-based study, so we really don't know, don't have any ideas. So I ran some simulation and figure out we need at least like 3,000 subjects we want to need us for these 130 chemicals. And I have put the analytical code online in GitHub. So if you check your paper, download the code and try to run the simulation yourself. And then we have some other analytical methods for analyzing the mixture. And there are people doing this review, so I'm not going to go into the details about those statistical methods. But most of them, they are more like about variable selection types of methods that you may have lots of variables, and you want to figure out those few variables, and they have a very high correlation with your outcomes. So, for example, the SO methods or something for the DSA algorithm. They call it the deletion, substitution, and addition algorithms. And then I want to show you a poster study of genomics after telling you about like the recent development about the study of the exposomes. So it's a study from Science. It talks about the exposure to the endocrine disrupting chemicals. So these are endocrine disrupting chemicals. They could be pesticides, they could be heavy metals, could be some other like perfluoro alkyl substance, things that you can find maybe in food, maybe in furniture, maybe in other different sources. Some of them, there may be some kind of a natural compound found in soybean. So, these chemicals they call it endocrine disrupting chemical because they mimic hormones in your body, and they bind to the hormonal receptors. And these are hormonal receptors. The things that is kind of important is that you don't need high dose exposure. These like some kind of like a signaling molecules to bind to this hormonal receptor, and then they will kickstart another cascade of biological mechanisms to amplify effects. So even though we have very low dose of this EDC exposure, it's important to know it may not be that kind of important you and me. Hormonal things are not as, but for pregnant women, for the fetus students, so these EDC exposure could kind of like have a long-term implication to things like neurological impact, like language impairment, like maybe autism, kind of like other kind of like cognitive issues. So this study is kind of about poster trial because really very comprehensive. Is more than just epi study. So they have the pregnant women, and then they measure about 20 EDC in the blood, and then they use some kind of biostatistical methods, weighted contact sum regression that I didn't introduce, to analyze the mixture, and then they figure out out of 10, so there are out of 20, there are 10 EDC seems to be strongly associated with language delay in the children. And then from this result, they try to synthesize those 10 chemicals, try to reconstitute that mixture with the observed concentrations and they create mixture, and then they have different concentration of that mixture, and then they move to do experimental stuff like testing these neurological cell, like the gene expression kind of experiment of gene set enrichment analysis to understand the influence of that mixture. And then they also have some kind of dose response toxicity test on a scalefish, and then the levies of the hawk, and to like another gene expression stuff, maybe some mobility tests to see how fast these fish larvae response to stimulus. And then after they confirm these toxicity effects, they go back to do assessment, and then they figure out about 54% of the pregnant women in their cohort, they are likely to be affected by the EDC mixture and have the children and language delay issue. So after talking about this poster child, so let's talk about the key approaches to measure the exposomes. So another science review can talk about like what kind of methodology we can measure complex mixture environments. So first we talk about some kind of chemical analysis, like using mass spectrometer to measure chemicals. And then so we have a targeted analysis, the way that we all know, like a very introductory stuff. Then we have the non-targeted screenings that we don't know what we are looking at, just try to measure everything we have in the samples. And then we have something in between, create the suspected screening. So there's no kind of like a universal method. It's more like always a trade-off between coverage and sensitivity. And you may think about like a flashlight. So it's powered by 4 AA size battery, and then you just switch on, and then you may turn it on the left hand side, and then you have the light field and a broad area that can be illuminated. Cannot go far, you cannot see far, and then you may turn the flashlight to another direction. You can focus the beam, and then for that one you can see far, but you cannot see broadly. So, the same idea here. So you have instruments with similar capability. So so you have to pick a choice, either the coverage or the sensitivity. And then targeted analysis, so we just have like collect the samples, do some sample preparation to enrich the analyte, and then you can spike kernel standard, get a calibration curve ready, and then measurements. Then simple stuff. So the advantages is that so we have absolute quantification. So we have the unit one line, maybe milligram per gram, milligram per milliliters. Then we can have confirmation of the compound. So you really know what you are measuring is the things you want to forget. And then for the non-targeted analysis, say the blood chemical, the blood metabolites. So it is a little bit more complicated. So you collect the blood samples, and then because your interest is only in the small molecules, so you want to get rid of the high abundant protein, like simple protein precipitation methods. Then you just take the supernatant and then shoot it into the mass spec, and then take it to like to measure whatever things and whatever signals, collect them. And you have the feature, and then each feature represents some kind of unit molecules in your samples. And then what you typically have is a table. You have molecular mass, you have the retention time, and other spectral information about this feature. It's not useful, it's not helpful, because it's not interpretable with this thing. We have to get a library, and then we have to do some matching, and then we may have some putative identifications based on the molecular mass. Then you can do something more like pathway enrichment analysis. Maybe you check the literature or what is the chemical come from, what is the biological effects, and how that helps you to interpret your research question in your studies. One issue here is that typically you only have about 15% of the known can be annotated, putatively annotated, and then more like 85% of the feature. There may be something useful there, but they are the factor of the exposome. So it's there, but we don't know what are those things, and interpret those things. And then suspected screening of kind of like in targeted and the non-targeted analysis. that so yeah, you have a few targets like the target analysis, but you don't have the standard. You just have the molecular mass information, the retention time, the spectral information of the things you want to look at, but you don't have the standards. And then you do the non-targeted measurement to scan, then you try to do the matching, which is one of them matching with a library that like maybe a million compounds. And then so there are other ways to tackle this like tracking complex chemical exposures. We don't know to talk about chemical analysis. We don't talk about chemicals. We talk about the effect size. We want to quantify mixture effects. And then we have different bioanalytical methods, like some cell life toxicity studies, or maybe some receptor-based analysis that we maybe some kind of a promoter assay that you have some kind of a chemical in the mixture bound to some kind of promoter, and that gives you a sync. And then the best thing is, of course, you can combine the two. And here I'm showing you a study that really combine the two and do something like interesting. So the target here is not human health, but total salmons. So it's a salmon fish that you can typically find in the northwestern Pacifics. That these are salmon fish. They go back to the stream, go back to where they are born and spawn. But every year, people can find a significant number of deaths of these coho salmon in the stream. So they don't know why, because there's no pollution. All right, and then these are the senior author of the paper, Agrobiotechnology. So he decided to do something called the effect direct analysis. That he collected the literature, and he know the literature has the toxicity and kill the coho salmons. And he used chromatography to separate the literature into two fractions and test which fraction has toxicity. So he keep repeating these nation steps to reduce the number of chemical, maybe like over 2,000 chemical that could be the culprit, down to 4. And then after more like a detective work, was able to narrow down to a single compound, 6 ppb, that has toxicities. And then after that, he has to do some more detective work to figure out where the thing comes from. And then he checked the literature, and then he found some kind of a cryptic document. EPA talks about this. Actually, they were added as an additive to the rubber tire to prevent the damage from the ozone onto the rubber. And is that compound quinone is the oxidative compound of the 6-PPD. And then 6-PPD quinone that's why it's coming from literature. So we have the car tire, and we do have this chemical release. We have oxidation in the sunlight, and then we have this 6-PPD quinone in the literature, and that kills the fish. So this is how you can combine these two methods of some environmental problems, regarding the culprit, the toxicity mechanisms, doing some kind of policy changing things, say maybe ban these 6-PPD, you can use it in car tire. And then let's talk about things in my group for the rest of the 15 minutes. So before I talk about the details, I have to ask questions about why measuring the exposome is difficult. We are using the high resolution mass spectrometry to measure this small chemical, the blood or the urine. So the figure on the left-hand side is from my PhD manuals, the report. I like this figure a lot. So you have the x-axis, the concentration, blood concentration of different chemicals, and then the y-axis is the abundance. And then we have 4 different classes of stuff. The blue one, the endogenous compound, things we have in your body. Then we have food stuff, drug stuff, that color, gray color. And the red one is the pollutants. And you can see there's a 3 lines can overlap the endogenous stuff, food stuff, drug stuff. So some of them, they have pretty high concentration. But you look at the pollutants, and they are a couple for all the lower concentration common stuff in your blood. So if you try to do some untargeted measurements, so it has limited sensitivity. So it may be good to explore maybe more than half of these endogenous drug stuff, food stuff. But it's very kind of like difficult to investigate the pollutants, the real exposures that we talk about, like exercise residues. And then to make the problem more complicated is that if you have low concentration, you may get some measurement about the molecular mass. You cannot get the spectral information because concentration is not high enough. You have very low quality spectrum. you can't really use it to some identification, conformation of the chemicals. So that's the problem here. Yes, you know what you want to look at, and that's why you can detect it. Okay, but if you don't know what you look at, and you want to do some exploratory analysis, and now we don't really have the capability to do that. And then, so I think that it's not really necessary to measure everything. So if we kind of like it's about the first question. So if we want to talk about health and diseases, human or plant or animals, then why don't we just try to focus on the exposures that we really expose it to, and the things that are really biologically active. Say, we look at the things in the interface in nature and nurture, biology and the environment. So that's where we should look at. And by what I mean is that I don't try to look at the chemical in blood, the chemical urine, chemical that really bind to some kind of biological molecules, that molecular interface. And then, so I tried to write down this concept properly, and I call it functional exposomes, together with my former PhD mentor, and Gary Roel and Craig is from Ronansky Institute of Sweden. So what a defining functional exposome is a subset of the exposome that it only contains this biologically active exposure. And then, so we have the concepts for how actually you can do the measurements. So I think we can do a general types of methods called affinity purification mass spectrometry methods that are basically like the samples, blood sample or water sample, environmental samples. And then, so you have some idea about the biomolecule receptors that you want to look at. You know the effects, you're interested in the effects, but you don't know what kind of exposure can affect these receptors. So you use them as the base, go to ELISA cake, then you load your sample to that. So after some incubations, so you have some binding, some exposure binding these receptors. Then we can do some elution and then send it to the high resolution mass spectrometry, and then to do the measurements. And there are different ways to analyze and the result. So, so one of the advantages is that talk about low dose exposure, and the methods itself, these affinity purification methods, it's actually like enrichment methods. So you are kind of like keeping the things you want to look at, and then you remove the things you don't have interest, and then that will help to address the low dose exposure problems. Then I think the most critical advantage is that so we become clear about the things we don't know. For example, we talk about the molecular mass of something 503.111 daltons. This is things come from my study, and I don't know what's that. I only know the molecular mass. And then someone from Sweden, they have a study and they figure out the same thing, 503.111. So maybe you may be happy that we found something consistent of a disease exposure. But wait a minute. So the molecular mass can represent more than one chemical, and that is typical case. So you may have 10 or 100 chemicals represent to 503.1. So we talk about the same molecular mass, but actually we may not be talking about the same thing, because the mass itself is not unique enough to talk about this kind of non-communications. So if we have this kind of molecular binding receptor information there, the receptor is getting some kind of a completely different information, because the molecular binding is about the structure, the shape of the thing. Molecular mass is about size of the things. You have two different completely parameters. So if you have something with the same molecular mass binding to the same receptors, then we have much higher confidence to say, although we don't know what are those things, we don't know the identity, we don't know the structure, but we know that it's the same thing. We can communicate, share the result, and collaborate. There will be no more dark matter. So we don't know the structure, but we know the effects of the unknown, and we are kind of having a confidence of the uniqueness of the unknown. And then, so I think about if we can do that kind of collaborative work, we may have something like the human exposure atlas, a reference map of exposure for each disease, like something I read recently from Nature, as a human biomolecular atlas program. So the things that we have to ask is that like conceptually possible if you have this atlas, and also is that really useful if you like talk about this atlas thing about exposure? I think the answer is yes, because the exposome is unlimited, it's growing. The molecular size is controlled by the genome. And the genome, unless you talk about evolution, the kind of scale, time scale of disease, like your lifetime, but it's not change. So you have a limited set of biomolecule, maybe a lot, maybe not easy to measure. You have a limited set. And then you are using a limited set of biomolecule, but the more like limited dynamic exposome. And then about the usefulness, I think very obviously that we can do some gene and environment interaction studies. We want to know kind of exposure affect the gene expressions, and then we know some kind of maybe drugs that could be possible about the genes, that receptor, and then all of that expressions. And then so I tried to think about maybe some other interesting receptor about the exposure. So, but I decided to look at something more interesting, something about your circulating antibodies. Because like in the textbook, when they talk about the immune system, the antibodies, they talk about the antibody binds to peptides, protein, microorganisms, neutralize them. But they never talk about small chemicals. And then I read a paper that they try to detect formaldehyde, but they don't actually use antibody to detect formaldehyde. They use the formaldehyde molecule to capture anti-formaldehyde antibody in the blood, like a proxy of exposure. And I think about so there may be some kind of circulating immune complex that they bind to these small molecules. So I can extract those molecules, and I can use the affinity purification methodology to understand what are the exposure, what are the things binds to the antibody. So I did a simple experiment using my own blood. So one treatment, one control, and I just look at the top 20 signal with the highest approaching between control. I have some good identifications, so it's not confirmed that, so it may be something else. Anyway, but we can look at this good result. Some of the things is maybe hard to interpret. Phosphate, I don't know what I can. And then, but we have something that may be drug, like for the class drug exposure. But the first one, I think is ramic, and then we have something for the pep no per the something called like a barbiturate or metagonant, factor for fernandez. So these things drugs. I don't know why I first get this kind of things. But what I think is looks like the methodology can generally good at looking at drug residual, maybe some kind of antibiotics to the lifestyle, some kind of residual exposure that is where this thing come from, and that their concentration, people don't really measure these things in I don't know whether this is a real thing, and I'm going to have my own lab. I'm going to do something here. And then that's last 5 minutes, some very important remarks about stuff here. Exposomes are kind of a life course exposure, large dynamic one entity. Development in exposomics, government, they're putting investments, keep putting investments. Academia, we are developing methods, sampling methods, measuring methods, analytical methods. And then the industry, they are exploring market for these concepts. And then good news is additive cells science, they like this kind of study because you don't find this study like in Nature, not in JAMA, not in General Medicine, but only in these two journals. Looks like they are kind of favor about this exposome concept. And then it's possible to measure the functional exposomes with some kind of affinity-based methods. And also prelim experiments I can able to recover some chemical from the CIC, and it looks like these body receptor is kind of good at capturing drug residual or maybe antibiotic, the kind of something kind of study, maybe good at using this approach. And then So the exposome is like one field. I started to go to the lab and then search the keyword and see number of citation per year. So each bar is the number of citations. And so we have these 3 different ohms, and these are going up exponentially, but they are not in the same scale. So if I overlay the metabolome to the genome, and then it's actually only halfway about the absolute citation per year. And if I overlay the exposome to the metabolome, it's just in the middle of it. There's still a long way to go. It's a promising field, 10 to 20 years, we produce the success of the metabolome and the genomes. And then, so the original vision of Christopher Weil of the concept is more like we look at the exposure, like in the genome. So sequence the genome, sequence ATC, like the exposome. The original idea is we measure this exposure. We don't exactly look at it. And then in this sci-fi faction, Star Trek, they have something called a tricorder, and then it's more like you press a button, and then you can do a body scans about the health of a person, or environment scans about whether it's the atmosphere is safe for these people to go to a planet. And then, so we don't have this medical device, long way to have these things. But I do have a dream that we can have something like a critical size, more like a sampler with 500 5,000 receptors. So we can prick the finger, get a few drops of blood, and then that device will do the sample preparation, binding, also preparation of the sample, maybe for a week something. Then we can send it back to a lab, do the measurement, and then for the lab, then we can have the measurement. We can get some calculation like the exposure about say you have this kind of environmental exposure. So you have maybe high risk of getting type 2 diabetes. And then you also get the medicine to cure the disease, but also prevent the disease and have some kind of drugs that bind to the exposure, so that exposure wouldn't bind to the receptor and affect you. And then one more thing, the final things. So there are like a review articles in Nature talking about genetic strategy to like improve crop yields or relevant to I think audience here. And because this plant is not human, so it's edited. Can't really do this kind of thing human. So they have strategy, genetic strategy to improve crop yields. Then in that box one, it talks about there could be development. Of course, a safe small molecule that deliver to the plant, like a drug to the plant, that if stress or during recovery, that may be some kind of like a prime the plant to tackle adverse events, maybe climate change, because we have more extreme environment, like the drought or high temperature, maybe alkaline environments. Then we can also study the microbiome and how they interact with the plant and improve the plant health. So I suppose many of these small molecules, the natural environment, that coming from these microorganisms, synthesized from these microorganisms. I think more like a typical example is some microorganism takes the N2 gas from the atmosphere to something like a nitrate for the plant that helps plant groups, and then use the two microorganisms in similar way to improve crop yield or crop health. And then there are also 2 recent science papers talk about they can figure out genes that helps to improve grain yield, and a G gamma protein that helps to cope with alkali stress. So these things are coming from GWAS study. So we can more like one step back, talk about, remember we have the P equal to G plus E, that kind of equations. That applies to human, that applies to animal, that applies to plant. So the plant health, plant of phenotype, crop yield are affected by the gene, also the environment. And then we have the GWAS, like one of these GWAS, figure out that related to alkali sensitivities. And then we can also have extra related to these plant study, animal studies, and identify the chemical exposure that can improve the phenotypes. And then the APMS, the affinity purification methodology, is a very general methodology. As long as you have more advanced knowledge in genomics, like this GWAS study, we know the gene of this like high impact journal. They are impacting genes. And then we have a target to use this purification to figure out that there are some molecules in the nature, molecules microbiomes that may bind to this receptor, regulate their expressions, maybe increase susceptibility of plants to climate change. And so that's all for me. And thank you again for Professor Lam inviting me out here. And I also thanks my active mentors. So I'll just talk about my PhD mentor. My study from here, undergraduate science program, and I was adopted LMG students. And I went to Baptist University to do a master, and then I went to Berkeley to do a PhD, and Taiwan, and Chung University, and then to a hotel the postdoc period. And I also thanks the who work with me, and there are many people, US, Stanford, Columbia, Europe, Karolinska College, so Asia, University, National Taiwan University, and finally I got some icons that icons of knowledge. So that's it. Thank you very much.
Hey, so before we get on to the questions, maybe we can take a photo together. So
Cool. Yeah.
Thank you.
Thank you.
So now it's open for questions and discussions. So, okay, Albert.
Okay, so
Please
thank
unmute
you very
yourself.
much for Professor Chung to give
sorry.
me a wonderful.
I mean,
Hold on, we cannot hear you.
Okay, Albert, can you try again?
Yeah, so thank you very much, Professor Chung, for a wonderful talk. I am glad that you mentioned Columbia because I also graduated from Columbia.
Oh,
but
cool.
yeah, okay, so two questions. One is that which are the commercial company, right? So I'm a patent attorney. So who is actually commercializing this wonderful line of research? That's my first question. And if this company, who is the leading company, or doing what, right? So that's question number one. Number two, I was in a lot of international, like, finance meeting and whatever. And so a lot of people saying that all these like VC fund and initial early stage investment is really at a very low end. But unless you put these two letters, it's called AI. So I'm using GPT every day, maybe like 5-10 times. And how would you think that all this AI is going to impact all your wonderful research about exposome? So these two are the very curious and very pressing in my mind.
Okay, thank you very much. We have the muted. Okay, so I'll just keep going. So for your first question, I mean, remember correctly, is about who's going to like commercialize the stuff in the company. So I would say that this still like the field is still in its infancy. Based on my knowledge, there's only two companies doing this stuff. The one is the figures I showed you about my exposome. So because they have some kind of like a silicon waistband sampler that they have a paper in environmental science and technology to demonstrate that it is something simple, effective to measure personal real exposure. That's why they try to develop a company to help people do some kind of measurement.
So Professor Chung, a lot of some of my
investors, it's only a sampler. It only works with SVOC and doesn't work with metals. And then so it doesn't do the measurement. You need to go back. And then for the rest of the company, they are more like from the different dimensions. For example, my PhD mentors, XY.AI, that company, is not doing molecular measurements. It's doing something very typical in like a large country like America, Canada, China. People have a address code and mailing codes, and then you can use that code to integrate different information. say you want to know the education level of an area, and then they have the American Chemical like Society survey. You can download the data, and because of privacy, they wouldn't give the address of the information. They will only kind of like hide out the address and tell you for that zip code, we have an average like education level, income level. And we also use the zip code to maybe do some remote sensing thing to integrate different things together. And then that startup company is about integrating the databases, having an exposome data warehouse that you can have like insurance company, maybe they are interested in what kind of exposure and helps to get the patients healthier. But because of privacy stuff, we cannot really share the address. And then, but we know they are like insurance clients, typical, and we can do some association. Maybe for the foreigner level, people they are more active. So we may have a less investment about persuading them to get a smartwatch and share the physical activity. If people are in other areas, maybe they have a lower education level, they have a longer working area, just too tired to go back home, just want to do physical activities. So you may focus their resources of the insurance company. Maybe if you insurance from me, then I will kind of like have the discount if you buy a smartwatch, and they will record like a physical activity levels. So they will keep you motivated person activities to reduce the health risk. And that's that company about. And then there are some metabolome company that they have to do metabolomics, genomics, but it's more about the effect size, and it's not actually about exposure. My like a simple answer to your question is we don't really have that kind of company. Maybe more advanced in the way that we talk about integration, simple integration, because that's easier. But for the molecular way, molecular side, no, we don't actually get that company. No, not startup company. Even my friend Jenna has a company, media market, is about the metabolism, not about chemical measurements. And then for your second question about artificial intelligence, machine learning. So that part is more specific about building the biostatistical model to understand chemical exposure. Say for people like me, when I was at Harvard, we don't have a lot left. We don't talk about chemical measurements. We just have the big data, like 50,000 people, 1 million people from this UK Valve and all of us demographic capture rate. We get the HIV stuff from Africa, India. So we had the outcome, the HIV, type 2 diabetes, a high glucose level. Then we have many questionnaire, that kind of measurements. Then we want to figure out which exposure to the outcome. And we have these machine learning things, AI things, like the XRAS. So they are kind of machine learning algorithms to help us to build a predictive model. So try to figure out which are those like exposure kind of statistically significant to outcomes. And then we have the predictive model, and then you can talk about maybe talk to the insurance company, scores, and you can talk to your patients, measure certain exposure, and some kind of questionnaire stuff you can ask your patients, and you can build a score and tell about their type 2 diabetes risk. And then my recent understanding of the deep learning things to the chemical side is try to interpret the dark matter from the targeted measurements. Remember, I talked about for these targeted measurements, you have the high resolution measurements about the molecule. You have a library matchings, and then have a putative measurements. But then if you don't have the library, and then you just have the spectrums, so you can't interpret the thing. It's just something there. And I know there are companies, they talk about they have a novel deep learning kind of algorithms, like a neural network that type thing, very complex. Right? They have lots of data. And then they try to figure out maybe some spectral fingerprint, and then they could able to predict maybe like the structure of a chemical, maybe some functional group of a chemical. So that kind of work. So I know that they have a startup company, but they talk about their own step creator. I have followed it, but I haven't really heard about anyone really buy the concept or trust them about kind of deep learning things about the mass spec, get some validated really mean something noise. Like these algorithm things, pass in, trash out, publish in, publish out. Yeah,
so Professor Lam, may I make one or two comments?
Yes, go ahead.
Right. So number one is that some of my investors, they like your term, Professor Chung, is the word infantry. Like meaning there's a lot of things they don't start out like small. So they would like that the two company when it's beginning very small. So maybe that ultimately nothing happened. Maybe ultimately 10 years later, they will grow exponentially. So if you know of any company, you're going to create a company, Hong Kong is very popular to create company, let me know. So we have at least like 5 investors. They say that, okay, if I throw in $1 million US, 20 years later, you see the 1 million go to 1 billion. So that is a great, I like the term infantry. So second term is about all this AI. With the AI, I love you when you talk predictively, but it's the overall, could it be generative? I think that the AI still have a lot of space to grow in your field, and that would be actually a very wonderful stuff. And so, but thank you very much. I opened up my eyes. Before your speech, I do not know what exposome is, and so I do some. I asked ChatGPT, what is exposome? And so, but thank you very much. So I'm very contented.
also good to know about this. Because I was thinking if I want to do a startup, I had to go back to America. Now I know I can do it in Hong Kong.
Thank you.
Thank you, Albert. Is there any other questions?
I have a question.
Oh, Sandra, speak louder.
Okay. Actually, I have a few questions, so I don't know if we have time for all, but one question is that I'm quite intrigued by the microneedle-based interstitial fluid extraction for drug analysis. So it almost sounds like Theranos, but it's actually true. So I'm just wondering, how do you ensure accuracy when measuring and extrapolating the data from such a small sample volume? That is the question.
Okay. So so for these like a high resolution mass spectrometry stuff, usually you need microneedle cell. So that microneedle things, so you there's a video, they actually they don't just keep then 20 minutes don't want like a drug optimize the methods. Then they have a suction and then so that generally will have microneedles. So that will have to do a measure by. So that's not an easy thing. So if you have a targeted measurement like this, the device, the instrument itself, it only takes a very tiny microliters. But it's your sample preparation methods that takes about. Like when I was at Hong Kong University, I do soil pH and soil pesticide measurements. I take quite a lot of soil because the concentration low. I do enrichment, kind of something that source that extraction. It's more like you keep with salt, extract it. And then you have more like maybe fuels of solvents. You dry it, evaporate, and then down to maybe something like a 1 mil something, like in something. So you can keep drying it maybe a couple of minutes. Then for you to extract it, understand that if your sample is like 10 microliter, your needle is 10 microliters, and then error rate could be really high. Then if you have maybe 1 mil of sample and 5 microliters, margin of error. And then you have put the sample in the vial. Actually, when you put it in the GC, only take a few microliter in vials. You can measure maybe 2,000 times. Just keep measuring. You have samples. It's not really a sample of a measure. And for these untargeted methods, you don't actually need. And for that one, they don't have the blood. They don't have the hormone. The thing that the mass spec can say, and you can just do maybe direct infusion, skip the micrometry, just send the things in the market.
And apart from that, I want to ask if there is any temporal study that is, let's say we have, let's say pesticide exposure. Is there any exposome study that measures that? How long does it take for you to be removed from that heated source in order for the human body to clean up the pollutants from the human system?
Okay, so it is actually about things with the pharmacogenetics and studying toxicogenetic things, like you have an exposure, talk about the half-life of chemical in your body, and then so these kind of methods that they can't nothing, they are not decided because like if you want to measure a lot of coverage, your sensitivity, your accuracy, your sacrifice. About these kind of half-life and highly kinetic things has to be very hard to measure, very high sampling, high accuracy to tell the numbers of a half-life. It's a number to have some kind of application. What company they have a similar character, they check the database and then tell you how long this one can hear from your bodies. So they have some like more like consequences. For this kind of exploratory analysis, everyone knows it's an exploratory stuff. So we just want to find some. So we will do follow up. So can't just say I have a theory on something and talk to a medical doctor. So this one is a cause. It's not that way. But everyone knows it's a exploratory. And follow up confirmation for the job. Like I showed you such as that. So we have the measurement and the statistical and the experimental thing that confirm the mechanism. And you get that's the kind of the paper that has typically like the EDA stuff. You also have the from the really difficult cost that.
Also, a final question is that while it's interesting to hear that there are some startups that are utilizing exposomes and also XROS, is there currently any government bodies that are interested in adopting this whole system exposome approach in regulating harmful pollutants or chemicals in their policy making?
No, not yet, not yet. When you talk about a policy, that means you have legal consequences. People are very careful. They don't want to be pioneers. Because you want to be the pioneer. If anything happens, that's your responsibility. Just like remember correctly, you are air pollution. They have the methods, and then you say if you say that industry using like some over the pressure, your EPA official will sue them because you are releasing some toxic over the pressure, like regular pressure. And if they know you are not using the method exactly as described in regulation, or say some step just shortcut, then they can talk to the judge. So this method is the one. The judge can really throw away the whole case. They don't want to do that. So this kind of recovery stuff, like it, they want it. When I have meeting, like these like one of the exposure with Duke University, there was like an officer responsible for the biology branch, and then openly talk about they like this kind of exposure. They want it. They want you to, but they want to reveal is not them. That's they talk about. They want us to research it to be more open-minded. When someone talk about exposure, about exposure analysis, research proposal, don't just ban it. Not to try to know. The purpose is so I also have a hard time when I have kind of like plan proposal. That's that kind of thing. So when I talk to the people, that's what they talk about. They talk about exploratory analysis. You want to get a grant? No, that's a bad idea. Don't be right. They don't know what it, and they don't like this kind of exploratory stuff. They want the money well spent. They want hypothesis to answer yes or no to hypothesis. So that's what some of the stuff here. But the government, they are very welcome to say, show you this NIHS, their strategic research plan. They even have that in 2012, like the very early beginning of the exposome concept in America. They want to talk about that. Yeah, keep doing this thing as a focus. Sometimes having this request for application. So it's not like the funding that like GRF. You write a proposal and then like the government, they have the money and they have some project in their mind. They want you to write something, just address the questions. And then for that one, they can talk about omics. They have the money, they have the full control. So they're trying to do that, but it is still a long way to go. Like this probably based we even don't have a single method. You talk about the genomes. I don't have to explain to you. We talk about ATs, molecular level. You don't talk about chromosomes, or the genetics. We talk about these lower level, molecular level. But we talk about exposome, people like me, we talk about chemical. People like public health, traditionally about like things like air pollution, things like income level, social stuff. What they talk about exposure. So last year I have a meeting, there's a workshop held by NIHS. I have a few hundred people on different things. And I have a sections I talk about these chromosomes. It was very difficult to communicate because I talk about molecular stuff. People in the section talk about high level things, like this is social stuff. They are exposure, they're different things. So I think unless one of the methods prevail, I hope that will be molecular stuff because it's not to be mandatory. Like health, human health, like piece of medicines, those regularly. Unless we have something to go about the technology, and I think you won't have some establishment of different government.
Okay, so I guess we have a long day today. So is there any other questions? So it's not right. So well, thanks Professor Chung again, and we will announce the next talk data. So yeah, there will be talks in the coming months. So this series will be continued. Okay, so good night and good morning, everyone. See you. Bye bye.
Thank you.